This is work we recently published in Nature Communications, and then there is some other unpublished work at the end of the talk. And today's topic is on something we just did perhaps right now before starting the lecture, which is food seeking and eating tasty, sugary foods. And our lab has historically focused only on fear and anxiety type behaviors. So this food seeking venture is a very new topic for us. So I'll also talk about how we got to this sort of issue in the first place. Okay, so because our lab is very interested in fear and escape and anxiety sorts of behaviors, one region in the brain we study a lot is this region in the midbrain called the periaqueductal gray. So aqueductal from being near the aqueduct and peri around. So it's this donut-shaped structure around the aqueduct in the midbrain. And this region has been shown by many people over many decades to be very important for panic-related escape behaviors, among other defensive actions. So here is one piece of work from the Branco lab showing that if you look at the neuronal activity of this region, you see that glutamatergic cells there are activated when the mouse starts escaping from a threat. So they're somewhat silent before the escape happens, then their neural activity increases during the escape, suggesting they might cause escape. And indeed, if you optogenetically activate the glutamatergic cells in this region, you see robust escape happening, shown here by these bursts of high velocity. And this was done with glutamatergic cells. So these and other works have shown that activation of glutamatergic cells in this region causes escape and also freezing and also other sorts of defensive behaviors. So then our hypothesis seemed very reasonable, which was that perhaps if we activate the inhibitory GABAergic cells in this same region, it would suppress these actions. So the mouse would escape less, freeze less, be less scared, less panicky, more relaxed, and so forth. So our prediction was that the GABAergic cells in this periaqueductal gray region, or PAG, would be encoding threat proximity or threat distance, and that the activity of these cells would suppress defensive actions elicited by threats of various types. So we first tried to record their neural activity by expressing the calcium indicator GCAMP in these cells. So this is an indicator that becomes more fluorescence in the presence of higher amounts of neural activity. So monitoring the fluorescence of this indicator gives us a readout on the neural activity of these GABAergic cells. Okay, so then we express the neural activity indicator in the GABAergic cells, and then we put the mouse in contact with two different sources of threat. So one here is the elevated plus base, which has these closed arms and these two open arms. So the mouse prefers to be in the closed arms because it's potentially less vulnerable to predators and things of sorts that might attack it. So the mouse is somewhat averse to going to the open arms. And then this aversion to the open arms is a very pharmacologically well-validated measure of rodent anxiety. And the other threat we put the mouse in was this predator. So the predator is a lab rat. And in the wild, the rat eats mice, so the mouse is very scared of being in the presence of a giant monster. So then this is the other threat to expose the mouse to. Okay, so we record the neural activity of these cells using fibrophotometry. And here is our heat maps showing in which location of these environments there was more or less neural activity, and higher neural activity is indicated by warmer colors. So you see in panel M here that the mouse kind of doesn't go very near the right, which is where the rat is. So he avoids the rat, showing mouse is averse, scared, etc. of the rat. But you don't see more or less neural activity near or far from the rat. And similarly in the elevated plus maze, you don't really see more or less activity in average in the safe or dangerous parts of the environment, which are the open arms. So it appears that these cells don't seem to have neural activity that's correlated to distance to these threats. So these threats don't seem to be encoded in the neural activity of these cells. So that made us sad because we didn't see our prediction. But then we thought, oh, perhaps the neural activity of these cells might not correlate with threat proximity, but perhaps if we optogenetically activate or inhibit these cells to perturb their activity experimentally, then maybe the defensive actions elicited by these threats would change. So we optogenetically activated or inhibited these cells and then measured a wide variety of types of measurements related to aversion of open spaces or aversion of predators, and none of the measures seemed to change, either with inhibition or activation of these cells, which then made us very sad because the whole project seemed to not be working, because the cells don't seem to be correlated with threat proximity. Their activity doesn't seem to be altering defensive actions induced by the threat. But we observed that although these hypotheses turned out not to be correct, it's not that the activations of these cells were doing nothing. So it's true that the activation of these cells didn't suppress defensive actions and escape as we thought, but something was happening because the mouse appeared a little more exploratory, and sometimes it will nibble on things. So I thought, oh, perhaps it causes some kind of eating behavior, and then we explored that further and saw that, oh, indeed, the activation of these cells seems to induce more food seeking. And this is what this talk is about this unexpected finding after we observed that our expected predictions were all wrong. And when we look back into literature, we see that indeed there's a lot of other sorts of work from previous groups that have shown already that there is a role for GABAergic transmission in this region that is implicated in food seeking. So more specifically, it was shown that there are many different regions like the lateral hypothalamus and central amygdala, etc., that project long-range GABAergic projections to the periaqueductal gray. So all of these projections are GABAergic. And then optogenetic activation of any of these GABAergic long-range projections to the periaqueductal gray seemed to induce hunting of crickets in mice. So we always tend to think of the mouse as being a prey and everyone wants to eat the mouse at all moments. The mouse is very scared, which is true, but the mouse also is a predator itself. The mouse predates little insects like crickets and beetles and little small things. So the mouse is also a predator, and then activating these long-range GABAergic projections to the periaqueductal gray seems to induce this hunting of insects. And this suggests that GABAergic transmission in this region might be important for this behavior, but the role of the GABAergic cells themselves that are living in the periaqueductal gray has not been explored. So this is what we decided to do. So the two of the main questions we tried to address were, if the activation of GABAergic cells in periaqueductal gray induces hunting of insects, and also does it induce hunting only of insects, or also perhaps food seeking of other types that are not prey-predator interactions, just regular food. And lastly, you can see here by this diagram that all of these previous works that implicated this region with hunting are kind of showing that inputs from other regions of periaqueductal gray, when activated, can cause this hunting behavior. So we want to see if there is some output, like long-range, bottom-up output from the periaqueductal gray to other regions that would also induce this behavior. So first, we recorded the neural activity of cells in this region that are GABAergic. And to do that, we expressed the calcium indicator GCAMP, again in the same cells. And we recorded neural activity using the UCLA miniaturized microscope. And then we exposed the mouse to 2 food sources throughout the whole talk. So one food source is walnuts, which is a very kind of calorically dense, fatty, natural occurring food, and to a cricket. So the cricket represents here the prey-predator interaction, which is a different type of food seeking, but it's a natural behavior that the mouse does. And we had never done any of this cricket hunting before, so we thought these mice will never hunt anything because they just stay in the cage and they're fat and they just eat their food all day. Why would they be hunting something? But then to our surprise, even without food deprivation, like a fully satiated mouse, if you put him in front of a cricket, within 20 minutes, essentially 100% of the mice figure out that I can hunt the cricket. And then after they learn to hunt the cricket, they permanently now are cricket hunters for the rest of their lives, and they don't forget that they learned to hunt. So this is a natural behavior the mouse does very easily. And then we record the neural activity of the cells in these two environments. So here are some fluorescent traces of the calcium indicator GCAMP showing periods of more or less neural activity. And these are some graphs showing the locations of cells in the periaqueductal grid that we recorded through this field of view. So the first thing that we noticed very remarkably was that when the mouse was eating, so doing chewing, swallowing, biting, those eating-related actions, there was a decrease in neural activity, which you see in these blue epochs. So both when it's eating the cricket or eating the walnut, there is a suppression of neural activity. And conversely, there's more neural activity in the pre-eating exploratory food-seeking phase, right? Because before the eating happened, there was kind of approach to food and investigation to find where the food was. So we see more neural activity in the approach to food, exploration, foraging part of the behavior, and you see less activity during the actual eating per se. And this was seen both in some example cells, like this cell in the cricket assay or this cell in the walnut assay, and it was also seen throughout the whole population of cells. The majority of cells was inhibited during the consumption, eating, swallowing, etc. So this also already suggested the possible role that these cells have in food seeking. Okay, so considering that these cells are inhibited during the eating, swallowing, etc., that suggests that the activity of these cells is not maintaining ongoing eating and swallowing. But instead, because the cells are more active in the pre-eating phase, it suggests the cells might be controlling these pre-eating behaviors, such as the approach to the food, the hunting of the cricket, and so forth. So the cells from this result appear to be more related to food seeking rather than the eating per se. And we could use decoders by using logistic regression decoders to decode the approach or food-seeking behaviors and the eating actions in both of the walnut and cricket assays, showing that these cells were representing these actions. And if you remember, in contrast, these cells were not representing distance to the threat, either of open locations or of predators. But these cells do seem to be representing these foraging, eating-related actions, suggesting that perhaps they have a role in this. And then to test that, we optogenetically inhibited these cells. So we expressed the inhibitory opsin arch. So this is an opsin that in the presence of green light will pump out H+ hydrogen ions, and then pumping out these positive charges will make the cell more negative, more hyperpolarized, so more inhibition of the neural activity. And we see that in the walnut assay, when we inhibit these cells, it decreases the amount of walnut consumed. And in the cricket assay, it increases the latency to get to the cricket, and it also decreases the capture probability of the cricket. So the fact that we see this increase in the latency to get the cricket suggests that these cells are important for initiating this hunting sequence and food seeking and so forth. So to see if the activation of those cells could cause that behavior, we then did the opposite experiment where we optogenetically activated these cells and then tried to see if the activation would increase these food-seeking actions. So here we see a video where there's a mouse here, then there's a cricket there, and this is a non-food deprived mouse. This mouse has normal access to food, he's not hungry, and even satiated mice also hunt crickets. And you'll see that during the laser off period, before we activate these cells, the mouse has some interest in the cricket, and the cricket is this little guy here. And then when we optogenetically activate this the GABAergic cells in periaqueductal gray, you see that the hunting behavior is very qualitatively different from the baseline action. Okay, so we then have the baseline behavior. So the mouse has some interest in the cricket, but it's not super motivated to catch it. And then when we optogenetically activate these cells, you see that immediately goes towards the cricket with a lot of motivation and catches the cricket and eats the cricket and is very happy with this cricket hunting ability. And we also have a video like this where we show that the optogenetic inhibition decreases cricket eating, but no one wants to see a video of the mouse not eating the cricket, so it just shows the activation video instead. Okay, so, and this is the group average data showing that if we optogenetically activate these cells, then the mouse shows a decreased latency to eat the walnut. So he approaches and starts eating the walnut more quickly, decreased latency to eat the walnut. He also consumes more walnut, and he shows a decreased latency to eat the cricket. So he approaches the cricket more quickly, and there's a higher probability of capturing and eating the cricket. So in both of these sorts of different behaviors, the activation of these cells seems to increase the food-seeking actions, leading to higher consumption. And here there is something to notice a little, which is that we show two effects, right? We have this increase in food-seeking effect, which is shown by the decrease in latency to get to the food. But there's also higher consumption. But if you remember from the previous data, there is less activity of these cells during the consumption. So the way we make sense of these two different pieces of data is that the natural activity of the cells is higher during the food seeking, and then it's inhibited during the eating. And then when we're activating these cells, we're kind of increasing this pre-eating activation that might be related to the food seeking, which causes increased food seeking and lower latency to reach the food. And then once the mouse reaches the food, then some other brain region or other circuit takes over to continue ongoing maintenance of the eating behavior, because these cells don't seem to be directly causing the eating behavior because they're mostly suppressed during the eating. So this is how we interpret this action here, where the activation caused both increased food seeking but also more consumption, because probably after the food seeking is done, then some other brain region takes over and creates the ongoing consumption. And we could also see that in some more like casual observations, that that's probably what's happening, because we also gave different sources of food to the mouse in behaviors that are not in this paper. So we gave, for example, little pieces of like apple. And then if you cut a little piece of the apple and activate these cells, then the mouse goes near the apple and then picks up the piece of apple. Then he does the food seeking, which is picking up the apple to see what it is, and orally explores the apple. Then he goes, I don't like apple. Apple is very boring. I won't eat the apple. So you could see that the activation caused the food seeking, which is I'll get the apple and pick it up and see what it is and explore it. But it's not fun, so I won't eat it as much. But if it was a kind of tastier food, like a piece of bacon, for example, or sugar or whatever, then he also went faster to the sugar and bacon and picked it up. And bacon is very good. I'll eat a lot of bacon. So the cells do seem to be causing indeed this food seeking, not exactly the eating. And that's something we're exploring more in detail in our ongoing work after this paper was done. Okay, so then we tried to kind of separate out if we could separately measure the food seeking without the consumption, because in this previous experiment, the food seeking and the consumption were linked because the food was right there. So if he food seeked more, he also would end up eating more. So we did this other experiment where we put the mouse in the box and then we put the food or an object kind of hanging from the ceiling of the box. So then the mouse can see the walnut, smell the walnut, but it's too high, so he can't reach and catch the walnut. And these are again all satiated mice, who are not super hungry to begin with. So the control mouse spends equal amounts of time throughout the different parts of the box, but the mouse where he did the optogenetic activation of these cells, he spends much more time directly underneath the walnut that's hanging from the ceiling, like much more time. And this is happening even though he cannot eat the walnut, right? Because the walnut is there and he can't reach it, but he's trying to catch it. So this shows that the excitation of these cells can cause more food approach and food seeking, even if you decouple that from eating the food. Okay, so this shows that these cells can cause more food seeking, and the activation also causes lower latency to pursue the cricket, because The mouse wants to hunt the cricket, but on its end, the cricket doesn't want to be eaten, so the cricket wants to run away from the mouse. So this chasing and running away and pursuit is an initial important part of this hunting interaction. So we wanted to see if the activation of these cells could cause pursuit of things, because chasing and pursuit is an important part of this hunting behavior. So we did this very unusual test called the ball follow assay, which looks like this. So here's the control YFP mouse, and on the right side, the mouse that we're optogenetically activating these cells. So what will happen is we will put a orange plastic ping pong ball on a stick, and then the experimental will move the orange ping pong ball around the box. So this is not food, it's not edible, and there's no training. The mouse has never seen ping pong ball. He has no association between this and hunting or cricket or anything like that. And what you'll see is that when we optogenetically activate these cells, it causes kind of pursuit of this ping pong ball in the optogenetically activated mouse, but not in the control mouse. So you see the control mouse has absolutely no interest in table tennis or other sports, but the mouse where you optogenetically activate these cells, this mouse is very interested in the ping pong ball and goes wherever the ping pong ball is going, which is sort of the initial behavior in the hunting process, where the mouse has to follow, chase, go after the cricket. So you see that when you optogenetically activate these cells, there's a high overlap between the trajectories of the mouse and the trajectory of the ball. In the control case, there's absolutely no overlap between the two trajectories. And that's quantified here in this ball follow assay, where you see that the activation optogenetically of these cells causes an overlap on these trajectories, which is kind of quantified here, basically showing that activation of these cells causes this pursuit of objects, even if they're non-edible, suggesting that these cells have something to do with this food-seeking, hunting, pursuit sort of behavior. And because it causes ball following rather than ball aversion, it suggests that these cells are causing food seeking due to some positive motivation, because he's not running away from things, he's running towards something and he's seeking more food. And that's an important kind of distinction in the feeding field, which is why is it exactly that activation of a circuit is causing more food seeking, more eating, etc. And there are many different reasons that that could happen. And some of them could be sort of in opposite emotional valence. Okay, so for example, one reason that the activation of some brain region could cause more eating is because that brain region causes more hunger. Like more hunger would cause more eating, but the hunger is aversive and the animals don't like to be hungry. So hunger would be a kind of a negative way to induce food seeking. But there's also ways to induce food seeking that might be related to some positive motivation, like some anticipation of some tasty reward in the future, something like that. So there's both positive and kind of negative valence actions and psychological processes that might induce food seeking. And this is, much more complicated than in our previous field of investigating fear behaviors, because there's only one sort of thing that can cause a fear behavior, which is some perceived level of danger, and that is always negative. So there's no positive thing that causes a fear behavior. But in food seeking, both positive and negative psychological process can cause food seeking. So this suggested that this might be a positive motivating process because it causes more ball following and more approach to the food. So to look at that, we did this real-time place preference test, which is basically you put the mouse in these two interconnected boxes and the mouse can freely go from one box to the other. Then at baseline, the mouse spends equal amounts of time in both subcompartments because there's nothing particularly happening there. But if we optogenetically activate the cells whenever the mouse goes into the right compartment, then we see that the mouse spends much more time in the right compartment, which is quantified on the right. So this shows that the mouse kind of likes this optogenetic activation, or this activation might reinforce ongoing behaviors or might produce some positive motivation or something like that. And there are some different sources of interpretations, but one possible interpretation is not that he is getting super hungry, because if the mouse got very hungry from this stimulation, then that would produce aversion, so the mouse would avoid that stimulation. And then we also did some other experiments showing that the mouse will learn to lever press to get more of this stimulation, which suggests that they sort of like or are attracted to, or at a minimum are not averse to this activation, suggesting that this is not causing hunger. So our interpretation then is that these cells are causing more food seeking due to some positively motivating process, such as the anticipation of getting some tasty reward or reward seeking actions, something like that. And because it's a positive motivation of some sort, we then wanted to see if this motivation would be enough to get the mouse to overcome some other negative motivation that decreases food seeking. And one of that negative aspects could be some danger created by electric foot shocks. So here we have a small box, and then the mouse is here, and there's a walnut that is on top of a shock grid. And then at baseline, the shock grid is turned off, so the mouse eats some amount of walnut, and there's no shock, so nothing happens, there's nothing to avoid. And then during the shock period, we turn on the shock grid, and the shock grid is very weakly active. So it's not the amount of shock that's used to create fear conditioning. That's a very intense, threatening shock, but this is more of an annoyance. So it's not some huge, painful stimulus. And then we see that the shock predictably decreases the amount of walnut the mouse is eating. But then we also see that if we activate optogenetically these cells in the presence of the shock, then that rescues this shock-induced decrease in food seeking that happens. So the increase in reward seeking that's created by activating the circuit is enough to overcome this negative shock-related aversive stimulus. So this is also a potential way to model compulsive food seeking, which would be food seeking despite the presence of negative consequences. And that's one behavior that can be seen in some human conditions like binge eating disorder, or also in some addiction-related disorders where the reward seeking happens even despite of negative consequences. Okay, so the next thing we try to do is to investigate if there were any long-range output connections from these cells that might be mediating this behavior. So first we just made these GABAergic cells in periaqueductal gray express the fluorescent marker YFP, and then we saw where do their axon terminals end up in, and we saw that the most robust axon terminal arborization was seen in the Zona incerta, which is a region in the subthalamic area that has been shown to be involved in food seeking by other groups. So if we inject this anterograde tracer here, you see axon terminals very densely and specifically labeling the Zona incerta. So this is also something interesting about the circuit, which is that these cells don't project approximately broadly to that whole region, but rather the projection seems to be very precise and targeted only to the Zona incerta and not to other nearby structures, as you can see from the green fluorescence here. And then we did some patch clamping to show that if you patch clamped cells in the Zona incerta, then you see a GABA being released, creating this monosynaptic inhibition in Zona incerta cells. So activation of these axon terminals does seem to release GABA onto the Zona incerta and create some inhibition directly in the Zona incerta. So this seems to be a real projection that's functionally connected. Okay, so first we tried to see when is it that this projection is active normally. So here again we made the GABAergic cells in periaqueductal gray express the calcium indicator GCAMP, which becomes more fluorescence, the presence of more neural activity. And those cells in the periaqueductal gray project to the Zona incerta, so that projection to the Zona incerta also eventually expresses enough amount of the GCAMP calcium indicator. So here you see axon terminals from GABAergic cells in periaqueductal gray that project the zona incerta. So this fluorescence here is from axon terminals of the periaqueductal gray. So now we can monitor the fluorescence of these axon terminals. So we are seeing not the neural activity of the cell bodies themselves, but rather which type of neural activity information is being transmitted to the zona incerta. And then we saw roughly what we see in the cell body recordings, the miniscope earlier, which is that there is an increase in activation of this projection during the approach to the food, and then the projection seems to be more inhibited during the eating part of the behavior. So there's more activation in food seeking and more inhibition of this activity during the eating, swallowing, etc., similarly to what we saw with the cell body data. And one possible confound of this data is that perhaps these cells are not inhibited during eating, but they're just kind of encoding velocity, and then there's higher velocity before eating and lower velocity during eating, and maybe that's what's happening. But that didn't seem to be the case, because when we recorded just in an empty box, then we don't see that when the mouse has low velocity or stops walking, there is a decrease in activity. So this decrease in activity is specifically related to some kind of consumption and is not seen in some other non-consumatory period of low velocity. Okay, so we next wanted to see if this projection to the zona incerta is activated only during approach to food or also during approach to other stimuli. So we put the mouse in this U-shaped box, and on one end of the box there's a walnut, and on the other end of the box there's a wooden object. And both the walnut and the wooden object are behind a wire mesh screen. So the mouse can see and smell those stimuli, but he can't eat or touch it or consume it. And surprisingly, what we see is that this projection to the zona incerta is more active, not exactly when he's near the walnut, but rather it's most active when the approach to the walnut starts, which is shown here by these warmer red colors. So you see that the reddest activity is not seen when he's the most near the walnut, but rather when this decision is made kind of to turn and go to the other chamber where the walnut is, which again suggests that these cells and this projection also is more related to the deciding to approach the food rather than being near the food or eating or so forth. Okay, so we then did the projection inhibition. So here we expressed the inhibitory opsin arch in these cell bodies of the GABAergic cells in periaqueductal gray. Then those cells send axon projections to the zona incerta, and then those axon projections eventually also express this inhibitory opsin. So in the presence of green light, the arch would pump out H+ ions, making the axonal long-range projection become more hyperpolarized and electrically inactive, and then not release neurotransmitters. So we can inhibit the neurotransmission in this long-range projection. And doing that, we see that the food seeking and consumption decreased in both of the food assays we tested. So inhibiting this projection caused a increase in latency to seek the walnut, an increase in latency to hunt the cricket, and also a decrease in the amount of walnut eating and a decrease in the amount of cricket captured. So this shows that the activity in this projection is necessary for food seeking leading to eventual consumption. So then next we did the opposite experiment, which would be to optogenetically activate this projection and then see if this behavior is reversed. And then indeed we see that excitation of the projection of GABAergic cells to zona incerta causes a decrease in latency to eat the walnut, a decrease in latency to catch the cricket, and an increase in the amount of walnut and cricket consumed. So the activity of this projection seems to be both sufficient and necessary to cause more food seeking, leading to eventual higher consumption. Okay, so then that made us happy because we found some long-range projection from the periaqueductal gray that caused these behaviors. And if you recall from the introduction slide, all the previous work identified long-range inputs to the periaqueductal gray that were involved in hunting only, but not in regular food seeking. So we showed that the GABAergic local cell bodies can elicit both food seeking of prey, like cricket, but also of non-prey foods, like walnut. And now we see a bottom-up projection from the midbrain to the subthalamic region that is controlling these actions. So our predictions on that end were observed. But then it also made us somewhat confused, because this result was in direct discrepancy and disagreement with some other very reasonably high replicated results in the literature. Okay, because our result here is that if we activate this projection from GABAergic cells in periaqueductal gray to zona incerta, this activation of this inhibitory projection causes more food seeking, more eating, etc. Right? Because this is a GABAergic projection from GABAergic cells in periaqueductal gray, so the activation of this projection is expected to release GABA on the zona incerta, which should inhibit the zona incerta cells. So this result is suggesting that a inhibitory manipulation onto the zona incerta is causing more eating, more food seeking. But then the problem with that is that previous people have shown that activation of the zona incerta causes more food seeking, more eating. Well, our manipulation is sort of expected to be a inhibitory manipulation, because GABAergic cells from PAG are releasing this inhibitory GABA transmitter there. And this is just one piece of data among others that show this disagreeing result. So basically, it was shown by this and some other groups that if you excite the zona incerta GABAergic cells, it causes an increase in food seeking, increase in body weight, and so forth. And that inhibition of the zona incerta causes less food seeking, less eating, etc. So we are kind of in disagreement with this literature. So then what we proposed is that, well, perhaps then this manipulation, perhaps it isn't really only inhibitory, perhaps it's inhibitory directly, monosynaptically, but perhaps it causes some polysynaptic disinhibition or some other slower network effect that increases the activity of the zona incerta cells eventually. So our potential explanation was that, well, we see that the activation of this projection causes more food seeking, so perhaps this is increasing the activity of zona incerta cells polysynaptically somehow. And then to investigate that, we did this somewhat more complicated calcium imaging, where we expressed this neural activity indicator both on GABAergic cell bodies on periaqueductal gray and also on GABAergic cells in zona incerta. So this result here is that if you activate GABAergic cells in zona incerta, you can get more food seeking. And the reason they did this is because the vast majority of cells in zona incerta are GABAergic. So it's kind of like the striatum where the vast majority of cells are GABAergic. So here too, essentially all the cells are GABAergic. So if you use a VGAT-CRE line, you can target almost pan-neuronally the zona incerta. So activation of GABAergic cells in zona incerta causes more food seeking. And because we're studying GABAergic cells in periaqueductal gray, you can use the same VGAT-CRE mouse to target the GABAergic VGAT expressing cells in both regions. So we express this calcium indicator, GCAMP, in cell bodies of GABAergic cells in PAG and on cell bodies of GABAergic cells in zona incerta, but contralaterally. And we did that because the connectivity between these two regions is unilateral. So if we do this expression contralaterally, then the activity of fluorescence that you're recording in one region is only cell bodies from that region, and it's not being contaminated by long-range axons from one region going to the other or something like that. Okay, so we see that when the mouse is approaching to the walnut, there's an activation of the GABAergic cells in periaqueductal gray, as we observed before in green. But we also see there's an activation of the GABAergic cells in zona incerta shown in yellow. And here there's two things to notice. So first is that both populations of GABAergic cells are increasing their activity during food seeking. And then that shows that, well, whatever is the effect of this periaqueductal gray GABAergic cells, they don't seem to be very strongly inhibiting all the cells in zona incerta, because while the green is going up, the yellow is also going up. So it can't be that the green is super robustly inhibiting all the yellow cells. So that's one observation. And the other is that in both cases, you kind of notice that the green cells in periaqueductal gray are peaking first. And then the yellow trace sort of follows it with some amount of lag, which is kind of in agreement with our idea that perhaps these GABAergic cells in periaqueductal gray are causing some kind of slower polysynaptic network effect in the zona incerta that eventually acts to increase their activity, perhaps by disinhibition, but perhaps by some other mechanism. But regardless of what the mechanism is, these data are not supporting the view that the GABAergic input from periaqueductal gray is robustly inhibiting everyone in the zona incerta. So then we did this experiment to combine calcium imaging with optogenetic manipulation to see if we optogenetically activate the cells in periaqueductal gray, do you see then an activation in the zona incerta GABAergic cells? So to do this, we express the red-shifted opsin carmine in GABAergic cells in the periaqueductal gray and recorded from the cell bodies of the GABAergic cells in zona incerta their neural activity with GChemp. And we saw something super remarkable, which is that whenever we do the optogenetic activation of the cell bodies of these GABAergic cells in periaqueductal gray, we see an increase in the neural activity in the downstream cells in the zona incerta, even though this projection is GABAergic. And then we turn off the optogenetic excitation, the increase in activity goes down, and it's very reversible and noticeable. So it's an effect that's super big, so you don't even need statistics to quantify this. But one possible confound here is that perhaps this increase you see in the GChemp fluorescence is not related to neural activity, perhaps is related to this optogenetic light excitation that's being put in, and that light somehow leaks into the zona incerta and causes some spectral interference. So perhaps this increase in activity is related to some cross-spectral fluorescence problem rather than real neural activity. So to get rid of that confound, we repeated the same experiment with the exact same mice, but now they're under isoflurane anesthesia. And then under isoflurane anesthesia, if we optogenetically activate the upstream cells, we don't see more fluorescence in the zona incerta GABAergic cells. And this isoflurane anesthesia is important because it shows that with the same light fluorescence and optogenetic activation light parameters that were used in the top example, we don't see this fluorescence increasing, which shows that this first result was due to neural activity changes. This was not due to light doing some complicated interference with the fluorescence, because we have the same exact imaging and light excitation parameters under anesthesia. And the main effect of the anesthesia is to suppress neural activity. It's not to change light interference. So this shows that somehow, likely polysynaptically, the activity of the zona incerta is increasing even when we activate this inhibitory transmission from the GABAergic cells of periaqueductal gray, which shows that our discrepancy has now been resolved, because these people have shown that activation of the zona incerta GABAergic cells causes more food seeking, and presumably we also showed that activation of the GABAergic cells in zona incerta causes more food seeking, and this activation can happen even in some complicated way involving the release of a inhibitory GABA transmitter. And of course, there are many potential synaptic biophysical mechanisms that could do this. So one possibility is that the GABA released by this projection is causing some kind of disinhibition, but there are also other possibilities. So another possibility could be that these GABAergic cells in periaqueductal gray, they release GABA as we detected in the patch clamping, but they perhaps co-release some other non-identified excitatory peptide, and then the excitatory peptide is the thing that's causing the excitation. Or it could also be some unusual example where the GABA is acting as an excitatory transmitter, which is observed mostly during development, but has been found in some strange situations in the adult circuits as well. So from this, we can't say exactly what is the biophysical synaptic mechanism that is causing this, but we could reasonably confidently say that the optogenetic activation of this GABAergic projection is somehow causing an increase in activity downstream. Okay, so our conclusions then were that the GABAergic cells in periaqueductal gray are more active prior to eating during the food-seeking foraging period, and the activation of this circuit seems to elicit both hunting but also food-seeking of non-prey food sources. And then this is the first work that shows that there are cells in the periaqueductal gray that are controlling the seeking of and consumption of non-prey foods. Previously, only hunting had been thought to be related to this region. And we also found that this circuit is acting to produce food seeking not by increasing hunger, but likely through some positive valence motivation. And then we also see that this activity is being relayed to the zona incerta, and then that projection from midbrain to subthalamic zona incerta is important for eliciting this food-seeking action. And lastly, our results show that this activation of this projection is somehow polysynaptically causing some excitatory network effect downstream the zona incerta, causing to create more food-seeking. Okay, so this is what we had in our published paper. So now we can talk a little about our more ongoing recent work. So we decided to explore more first, kind of what types of foods would this projection of this circuit control food seeking of. So here we have two sources of food, the walnut and the cricket, and both of those are kind of very valuable food sources for the mouse, because the walnut is very fatty and tasty, and the cricket is difficult to hunt, but they still hunt and eat a lot of crickets even if they're satiated, suggesting that it's worth this effort of hunting to get this food even though he's satiated. So they're both kind of more high value foods for the mouse. So we wanted to see what happens if we give the mouse some low value food and high value food. So here we have an example of the mouse which is satiated, and he's exposed to, on the left, the mouse chow, which is the food he eats every day in his cage, and on the right, a piece of chocolate. And then he's not super hungry, so he's not eating anything in particular because he's a satiated mouse. But you see that when we optogenetically activate these cells, he eats the chocolate but not the mouse pellet. And we also quantify this and see that the activation causes much more eating of these tastier foods than mouse chow or other less palatable foods. So this is quantified here, where we expose the mouse on the first row to kind of healthier foods that supposedly are very good for you and have very many nutrients, like broccoli and carrot and cherry and bell pepper, so vegetables. Then the activation of these cells causes the mouse to eat more of these vegetables, but the effect size is somewhat small. Like it causes him to eat just a little more broccoli, a little more bell pepper. But when we get the same satiated mice and expose him to more caloric, tastier foods such as cheese or chocolate or sausage or sugar, then the amount of food eaten is on average much higher than the amount eaten for broccoli or bell pepper and such. And we also see this kind of correlation of the baseline preference of the mouse for a particular food and how much increase in food or eating we see of that particular food. Okay, so for example, the mouse doesn't have a lot of baseline preference of eating carrot. So if you just have a naive mouse and give him some carrot, he'll eat very little carrot. And if you have some naive mouse and just give him some cheese, he eats a lot of cheese, as shown in many cartoons. So he has more preference for cheese than a cherry naturally or carrots naturally. And then the optogenetic effect is correlated with that baseline preference, showing that the activation causes a higher increase of eating of the kind of more naturally preferred foods. And this is also something that suggests that these cells are not causing hunger. So like the first finding was that the cells didn't induce an aversive state. And the second is that the activation causes them to eat preferentially more pleasurable foods. Because if it was causing a lot of hunger, then he would eat a lot of whatever food is there if he's hungry, and this activation causes him to be hungry. But instead, this suggests that the circuit might be more involved in some kind of like value-related, pleasurable food seeking. And if you think of that, like there are entire categories of food that we eat not to quench our hunger, right? So for example, if you're super hungry because you haven't eaten for a long time, then you don't go, I'm very hungry, I'll eat a lot of ice cream. Like that's a very strange behavior. So for example, ice cream or desserts are eaten almost exclusively by people that are not hungry, right? If anything, you already ate a lot of the regular food, and then when you're already stuffed, then you go, I'll eat this ice cream now, which is typically done not out of hunger. And we think this is more what this circuit appears to be tapping into. And we also did this experiment to show if these cells had some kind of experience-dependent change in their effect. Okay, so we know from this data that the optogenetic activation will cause them to eat more of the food that they already seem to like more at baseline. So what about if we had some food that was tasty but didn't have any nutrition, such as saccharine? So saccharine is sweet, but it doesn't actually have any calories. So we first expose the mouse to saccharine, and the mouse never has seen saccharine before. Then if we activate these cells, it causes some amount of, like a relatively large amount of saccharine eating because it's sweet, so he's happy with the saccharine. So the optogenetic activation causes more saccharine eating, just like it causes more sugar eating. Okay, so then we decided to teach the mouse that the saccharine is not nutritious. And you might ask, how do you teach a mouse that the thing is not nutritious? So the way we did it is we food deprived the mouse, so now he's hungry, and then he'll want to eat whatever because he's very hungry. So then we gave the hungry mouse some saccharine and waited 4 hours before giving the real proper food. So the mouse then experiences that I'm very hungry, I ate a bunch of the saccharine, and then 4 hours later I'm still very hungry. So the mouse learns that saccharine is not nutritious this way because it continues being hungry. And then if you get this mouse that has now experienced saccharine to not be valuable metabolically, then there's no effect afterwards. If you get the naive mouse, the optogenetic activation causes him to eat saccharine. If you get a mouse that understands that saccharine doesn't help with quenching hunger, then the mouse doesn't eat it because it has no value for him. And it's interesting because the saccharine is still sweet, so it still has this taste value, but it doesn't have any learned metabolic value. And then in the fifth exposure to saccharine, this effect goes away. But in the fifth exposure to sausage or cheese or the other things, the effect is still there because he doesn't learn that sausage has no nutrition. Okay, so this suggests that these cells are very involved in not just eating tasty foods, because saccharine is tasty, but also eating foods that are perhaps more valuable to the mouse according to its experience and learned episodes. Okay, so we then try to see if the activation will cause them to eat foods that are being dynamically devalued. Okay, so the way it works is we first get a mouse that is under normal conditions, and then if you give him cheese and activate these cells, it causes a lot of increase in cheese eating because the cheese is valuable. But then we can devalue the cheese, and if you just give him cheese overnight, so he eats a lot of cheese, then he no longer feels the cheese is valuable, and in that situation, the optogenetic activation doesn't cause him to eat more cheese. Or if you do the opposite where you, so here you see the optogenetic activation at baseline causes him to eat cheese and chocolate, But after devaluing the cheese, because he ate cheese all night, then there is no effect of cheese anymore. The activation of the cells doesn't cause them to eat this previously valuable cheese because now he ate cheese all night, but it still causes him to eat more chocolate. And the opposite happens if you do in the opposite direction. So again, at baseline, the activation causes him to eat both cheese and chocolate. But if you devalue the chocolate but give him only chocolate overnight, then the activation doesn't cause any chocolate eating but causes cheese eating. So this shows something more interesting, which is this circuit is not simply causing eating of tastier things like cheese relative to broccoli, but it's causing them to food seek and eat more the foods that are more dynamically valued at that moment, right? Because if you just had ice cream for 24 hours, then you probably wouldn't want to eat more ice cream tomorrow. So these cells are very sensitive to what is the recent experience of the mouse. And most recently, we also found another cool effect, which is that, remember in this video here, we showed that the optogenetic activation of these cells causes them to eat chocolate but not mouse chow. And we saw that here too. So, and this was a little confusing for us. So we saw that the activation of the cells caused him to eat a little more broccoli or carrot and etc. So he ate a little of of everything at least, except of mouse chow. Like the optogenetic activation never made him eat any amount of mouse chow, even though it caused him to eat everything else by some amount. So then we thought that perhaps that's because the mouse chow is sort of always devalued, because the mouse chow is there all the time for my whole mouse life, so the mouse chow is devalued. And then this is very new data, so it's not plotted yet, but we did this experiment but using mouse chow. So now at baseline we see that the optogenetic activation causes him to eat, let's say, cheese but not mouse chow. But then if you give him cheese for like 24 hours and no mouse chow, then the mouse chow kind of becomes valuable because that's a different food from the cheese. And after that, the optogenetic activation causes eating of chow but not of cheese. So you can kind of revalue the mouse chow by not giving him that. And again, these are not food deprived mice because they could have eaten cheese to infinite amounts. So we can both devalue foods like cheese and also revalue foods like the mouse chow, and we see that in every occasion these cells seem to be causing a consumption and seeking of these more valuable foods but not of the less valued foods. So I'd like to thank all our collaborators and especially people in the lab that led this work, like postdoc Fernando Reis. So also like to thank our funding sources. And this is probably a type of slide you've never seen before. So this is a normal slide of people that help. So then we also have negative acknowledgement slides for people that refuse to give us any money. So this is a negative acknowledgement slides for these foundations. Because what will they do? Give us even less money, so negative acknowledgement. And then thank you for coming to the talk.